Okay, so we were done with the topic that was terpenoid. Now let's move towards another topic, that is nucleic acid. Okay, so there was a first reported scientist, or you can say that the scientist was physician. The name of the scientist was Mr. Okay, so basically he observed the chemical they were the name of the scientist. Okay, and he observed the chemical composition of pus cell. What is pus cell? Pus cell is a cell in which you can say that the microorganism are present in that liquid. He observed the pus cell, and that pus cell was of course different from the normal cell, so he named the pus cell as a nuclein. Okay, he gave the name nuclein. He further studied the chemical composition and nature of the, you can say, the pus cell, and further he he observed the acidic properties with with the nucleus of the pus cell. And then he observed that the acidic property is there. Then further he renamed the nuclein as a nucleic acid. Okay, so the pus cell are basically what they are, the white blood cell. Okay, and if you're talking about, so they are not a protein, lipid or carbohydrate, they are basically a biomolecule, and he renamed as a nuclein. But he isolated more, and he was like, it was located in the nucleus, and the basic structure, or you can say the chemical nature of nuclein was renamed by nucleic acid because of the acidic properties. Okay, so acidic nature. Okay, so which acid? That acid was the phosphoric acid. Okay, now let's talk about the nucleic acid. So further he investigate the chemical nature of the nucleic acid. He further observed that the nucleic acid basically consists of the pentose sugar, nitrogenous base, and phosphoric acid. Okay, pentose sugar, as you guys all know, that this is a 5 carbon sugar, and nitrogenous bases are the bases which is attached with the pentose sugar, and phosphoric, you can say that's phosphoric acid. This is a acid that is present in the nuclein. Okay, now let's talk about some points, or you can say that nitrogenous bases. About nitrogenous bases, let's talk about nitrogenous bases. Nitrogenous bases. Okay, so nitrogenous bases are two type of bases. One is purine base, second is pyrimidine base. Okay, so in purine base, first of all, the first thing, that purine base consists of two rings, and pyrimidine base consists of one ring. Further, he studied the purine base, and he observed the nitrogenous bases in purine base, that was the adenine, and guanine. Okay, and you guys can say that this. Okay, first of all, one thing more, that the 5 carbon sugar he observed the pentose sugar. But one thing I want to tell you guys is that pentose sugar there's 5 carbon, but in pentose sugar he observed that the nucleic acid are having the two types. One is the deoxyribonucleic acid, which is, you guys all everybody knows, that is the DNA, that is the genome, or you can say the nucleic acid, or you can say the nucleic acid or genome of our body. Okay, second one is the, you can say second one is the ribonucleic acid. The formula of the DNA, that is the C5H10O6O4. This is the formula of the DNA. If you're talking about RNA, so RNA is having C5H10O5. Only there is a difference between the one oxygen. That's why DNA is called a deoxy, means one oxygen less, and this is known as the ribonucleic acid. Okay, so these both are the nucleic acid. Okay, and in the next video we will be talking about some more things, but first of all let's talk about we are done with the pentose sugar. Now let's talk about the nitrogenous base. So adenine and guanine are basically the nitrogenous bases of the purine base, and these are purine base are of 2 rings, and this is of 1 ring. Okay, and pyrimidine is basically consists of 3 bases, that is cytosine, thymine, and uracil. Cytosine, thymine, and uracil. Okay, now let's talk about the structure of these bases. Okay, so if you're talking about the structure of the bases, so the structure of the bases, let me tell you that first of all let me make the structure of the purine base. The purine base is made up of 2 rings. Carbon, 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 carbon. Carbon, 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 carbon. This is hexagonal, and this is the pentagonal. Okay, in every purine base, the first one ring will be hexagonal and the second one will be the pentagonal. Second thing, that the amine group, that is NH2. If amine group is at the upper side of the hexagonal ring, or you can say pentagonal ring, it is important that it will be on the hexagonal, but in some cases it can be in the pentagonal as well. So if this is present in the hexagonal, so NH2, so that is called the adenine base. Okay, when the NH2 is at the upward side of the hexagonal ring, so that can be the identification of the NH2, that is adenine base. If you're talking about the guanine, so guanine is also having a 6 carbon ring, and second one is the 6 carbon, 5 carbon. So if the amine group is at the left side of the hexagonal ring, So that can be the identification that this is the guanine base. At the side of this, you can say this guanine base. Okay, we are done with the purine base. Now let's talk about the pyrimidine base. So pyrimidine base, this we are talking about general bases. Okay, so this is a hexagonal, and they consist of one ring. This is the pyrimidine base. Okay, so you can say that this is a 6-carbon ring, and they all will be the hexagonal. Okay, if the methyl group is there, methyl CH3, the methyl group is upward side and one base that can be known as thymine. If the amine is there at the upward side, that is called the cytosine. You can say that in adenine there was also amine in the upward side, but you can see the difference of the rings. The pyrimidine consists of one ring, and here the purine consists of two rings. If there is a one ring and two rings, so you can easily identify what is adenine and what is cytosine. So this is cytosine because they are having NH2 at the upward side. Now let's talk about the uracil. What is uracil? Uracil is basically present in RNA. This is not present in DNA, and you can say that this is the uracil, and uracil is having no methyl group, no CH3 group, and no, you can say that no amine. You can easily identify that if there is no sign of the methyl and amine, you can easily identify that as the uracil. Okay, so now if you're talking about some questions, that is, so as you guys all know that in everybody knows that in RNA there is uracil, but in DNA there is no uracil. Okay, DNA is having adenine, cytosine, thymine, and guanine, and in RNA there is a nitrogenous bases, that is adenine, guanine, cytosine, uracil. Okay, thymine will be replaced by the uracil. Okay, so the question can be raised that in which of the following the radical have two purine bases. So the answer will be two purine bases. The answer will be adenine and guanine. How? Because the purine bases are adenine and guanine. If the question is that the methyl group is present in which nitrogenous base, so we can easily identify that the thymine is having the methyl only in all the nitrogenous base, so the answer will be thymine. If there is a question that no CH3 is present in which nitrogenous base, no CH is present in which nitrogenous base of pyrimidine, so that the answer will be cytosine and uracil, because thymine is having the methyl group. Okay, and many questions can be made from this one. Okay, now let's talk about some structure of the nucleotide. So first of all, one thing, that the monomer of nucleic acid is monomer of nucleic acid is nucleotide. Now what is nucleotide? So in pentose sugar, okay, At carbon number 1, if there is a nitrogenous base, and at carbon number 5 there is a phosphate group, that is called the nucleotide. Let's see the composition of nucleotide. Composition of the nucleotide. First step. First of all, there is the first step. Okay, first step is that there will be the pentose sugar, 5 carbon. In first carbon there is the nitrogenous base, so that is called nucleoside. This is a nucleoside. And if there is a carbon ring structure, here will be the nitrogenous base. Here if you are attaching the phosphate group, this is the second step, and this is called a nucleotide. Okay, nucleoside. In nucleoside phosphate is missing, in nucleotide phosphate is there. Okay, clear? So now the question can be made that during the formation of the nucleotide, in first step, what is the first step is formed? So you can easily write there is the nucleoside form. If the question is that in nucleoside nitrogenous base is attached with which ribose sugar carbon, you can easily say first number carbon. If there is a phosphate group, so you can say that carbon number 5th. Okay, so that's the difference between nucleotide and nucleoside. Okay, so the second step is the nucleotide, and nucleotide is what in which the nitrogenous base and phosphate group is there, that is called nucleotide. Okay, now let's talk about that. If there is that in the next videos we'll be also learning about ATP. So what does ATP means? ATP means that there will be the nucleotide but 3 phosphate, adenosine triphosphate. Nitrogenous base can be whatever base can be, and nucleotide whatever. If there is uracil present, so that can be easily identified that this is RNA. And if there is adenine, guanine, cytosine, thymine, you can easily identify this is RNA. And if the nitrogenous base that adenine, cytosine, and you can say that uracil, guanine, so that you can easily identify this is RNA. Okay, RNA nucleotide. And this nucleotide make the polynucleotide. Polynucleotide can be RNA as well as DNA. ATP. What is ATP? ATP is the currency of the energy. What does ATP? The full form of ATP. So ATP full form is the adenosine triphosphate. Means that there will be the adenosine nitrogenous base and triphosphate, 3 phosphate will be attached there. So in the nucleotide, if there is a nucleotide and the nitrogenous base is adenine and phosphate is 3, in the chain of the phosphate 1, 2, 3, so that can be called as the ATP. In the next video we will be just talking about this. Our topic is also will be coming ATP. I'm just giving the overview of this. So today we have just learned about the nucleic acid. Okay, in the next video I'll be telling you that the structure of the nucleotide, that how the carbon to carbon is attaching, how nitrogenous base is attaching, how phosphate is attaching. So today we have just done with the nucleic acid. So in the next video I'll be telling about the fully structure of this.